Well, jackhammers are not the first and probably not the second thing you think of when you think about cancer research, but tiny versions of a jackhammer might just be the answer scientists are looking for to help with the fight against cancer. Here to discuss this new treatment, we've got Professor Jorge Seminario. He's a professor in the Artie McFerrin Department of Chemical Engineering at Texas A&M and the study's co-author. Professor, thank you so much for taking some time to speak with us today.
Oh, thank you, Alex, for the invitation.
All right, so let's start with this. How does chemical engineering and cancer research intersect? Where does your line of research begin to overlap with that?
Well, yeah, this is a very, very special situation. We have three teams here. One of medical doctors who actually do the experiments in mice and in vitro, and the group of chemists, those chemists who develop the molecule, who synthesize the molecule. And then we have the computational quantum chemists, like me, or physicists, who actually analyze the molecules and check if the molecule is going to do what they want them to do. And that was our work at Texas A&M. We took those molecules, the so-called molecular jackhammers, and jackhammers are named because they actually do that. They hammer the surface of a cell until they break it. I mean, they crack the cell and decompensate the cell, and then they have to die because it's totally decompensated. You open the whole protection, the cover, the shell of the cell, and dies. So these molecular jackhammers have to be designed in such a way that they operate when we want them to operate. They shouldn't be working anytime and any other time. They shouldn't be operating in other cells that we don't want to kill. So they have this feature that you can activate them with infrared radiation, what is so-called near-infrared radiation. Infrared radiation is a radiation very close to the red color, but a little less energy, and therefore is invisible. You cannot see it, but with a special instrumentation, you can actually see it. That's what they use in several protection systems, safety systems, to see if somebody is crossing those red lights, those infrared lights. Well, this light is not that so energetic that is not able, for instance, to kill our cells. That's the problem with X-rays. When you send X-rays, you kill the cancer cells and you kill every cell that is in the way. This radiation actually don't kill any cell, okay? It's even weaker than ultraviolet radiation, much weaker than ultraviolet radiation. So with this light, we are able to excite, the molecules kind of go to an excited state, and in that state, when they vibrate, they are able to actually disturb, to destroy. It's like the name say, the hammering, the surface of a cell. If this cell is cancer, produce cancer, it simply is being killed at that point. The great advantage is that you can point this light to the specific place where you want to kill the cells. Okay, if you don't have cancer cells in some place, you just don't put the light on that place.
So Professor, if I can here, how soon? I know this was effective, 99% effective in vitro, 50% successful with the mice. How soon are we to seeing this for humans?
Well, that's another problem, a more difficult problem, because it requires a lot of paperwork, go through the standard procedures in order to have some new medicine to be tested in humans. Mm-hmm. You know that, yes, you see with the vaccine was something quick, but still it took one, I guess, year or 2 years. But usually it takes from 5 to 7 years for a new drug.
Well, we will have to look for it. Professor, we have to end it there. I appreciate your time coming on. We will have you back and hopefully talk about the advances in your research that you continue to make. Thank you so much for your time today.
Okay, thank you very much.